I do have one, since my perception is that you have developed also an important number of phase 1 and 2 clinical trials linked with this initiative. So could you explain us a bit with?
What, in fact, we still have to make demonstration that, at least to the Ministry of Health, that all these investments are useful, and there was doing that. So at this point, what we decide is really to make the demonstration that going to NGS, which will cost a lot, at least at the beginning, is useful. So really our goal was to demonstrate through a randomized clinical trial that it's better to have these information that to avoid them, to avoid their knowledge. It doesn't cost a lot. And I really believe that, and I know that very few randomized trials are done in this setting. But I know that if we demonstrate that, it will open further investment and further input of different people from bioinformatics and everything. I really believe that everything is linked. The training of doctors is at this point insufficient for this, for what we call personalized medicine. It's not so complicated so far because we have only a few number of drugs, no more than 20 at this point. But in 4 to 5 years, we should have 50. So you have, at the same time you develop the biology, you have to think about the training, you have to think about drug combinations, and you have to go forward at the same time for all these points.
Thank you for your presentation. I'm Dr. Salazar from the Catalan Institute, and I was wondering what are the pros and cons of a whole exome approach versus a more targeted approach like, I don't know, 70, 80, 90 gene panel that will be those genes that are more likely to have targeted therapies in the near future in clinical trials?
In our mind, I mean, the first step is to go through to panel of genes. But I really believe that when you will have a lot more drugs available, and you will think about the combinations of drugs, to have an overview of all the metabolic pathways should be of interest to make the decision, and not to be fixed on specific on specifics. And also, what I can tell you is that when you do a targeted panel, identified panel, you go through probably 95 of all the mutations, and but you don't address the rare mutations, and that's a pity. And you have to deal with these 5% or less than 2% abnormalities that you don't take into account when you have a 100 or 200 gene panel. Yes.
Thank you for sharing the experience with us. I have a question which is probably a little bit too much technical, but if you use for these 1,000 patients, have you in the plan is do a panel and then the exome, or only the exome?
Okay.
And the question is related with the depth of the quality of the analysis. We are people that are usually requesting NGS analysis for a panel of genes, requested from our labs a depth of 1,000 reads. And then to do an exome of 1,000 depth is impossible, technically. I mean, you can do it, but it's very expensive. So my question is, which will be the clinical threshold that you will put in the analysis for the exome?
Okay. Consider that the two first, I mean, you have two different ways of considering that. I mean, first one is research, and the other one is clinical use. For research, obviously, we will need to go through exomes, probably further than exomes, consider all the aspects of genomics, including whole genome sequencing, if you want to make a good understanding of what are the cancer diseases. That's the first point, and probably what we have learned a lot during the last years about. You may know. I mean, you are participating in Spain with a lot of success in the ICGC consortium, and you know that at the point we have been. I mean, the consortium has analyzed 35,000. Specimens, and now we know that we will have to go further to make major understanding outside of these first 35,000 patients. So this is research. For the clinics, it's completely different. We have to consider that panels, according to the drugs you have at disposition, is enough. But it's a pity also to consider that you're not doing research on patients at this point. So we are in the process of initiating these protocols with gene panels and progressively address exome and probably whole genome in the future.
Exactly. My question was exactly referring to this point, because I will be afraid of doing an exome for clinical, because I will probably miss a lot of mutations that are not, enough represented in the tumor biopsy.
Yeah.
Fabien, first I'd like to congratulate your team for the effort that you've placed into solving pushing forward a very difficult problem. Can you tell us which are, I think strategics is one of the major problems in this whole thing. Which are the 4 or 5 major strategic hurdles you have found in implementing this program? Where are the difficulties?
In fact, there was no difficulties, because I mean the difficulty is to what I can tell you, the difficulties is to have the money. Okay, so money is not complicated, because I mean this program started with 2 million euros, which is almost nothing. The cost of the whole program at this point in France is 12 million euros, which is not a lot compared to what is done. With the drug. I've showed you some of the impact of prescribing gefitinib. So the biology doesn't cost a lot; it costs almost nothing. So that was not a difficulty to show that. The main difficulty was probably to have working together pathologists and molecular biologists. You may think about that when I go out, when I'm leaving. Then that was. After that, I mean, you have to convince that you spare money to the authorities, that what it costs is nothing compared to what is spared, and this was okay. Apart from that, I mean, you have to convince that cancer is a drama, and that the politics have to consider that I don't know the data, but they should not be very different as we have in France. But it's one of three person who is facing cancer sometime in the life, including those of politicians.
So what we wanted to hear from you is that this is easy. Thank you very much, Fabien.
Merci, thank you.